In this lab, we use peroxidase enzyme. This enzyme is found in many plants and animals, but what we have used in this lab is extracted from turnips. The function of this enzyme is to oxidate peroxides, specifically hydrogen peroxide. Hydrogen peroxides are formed as byproduct of many biochemical reactions in living organisms. However, it can cause damage to the cell. Therefore, peroxidase plays an important role in reducing these peroxides to form water. The structure of this enzyme is very large and complex, and it involves many multiple folds. And also, it has iron ion attached to its active side, which is working as cofactor. To start, we got 4 clean test tubes and a box of cuvettes. We also got 4 pipettes and labeled each one to not contaminate the others. This is a table that shows the amount of each chemical that we have put into the test tubes.
Using the spectrometer to adjust the wavelengths to 500 nanometer after blanking it. Mixing tubes 2 and 3 into a cuvette and place it into the spectrometer and taking the reading every 2 seconds for 2 minutes. We do the same with tubes 4 and 5 after mixing them into a cuvette. We then load at the data and choose the amount of extracts to use in the rest of the experiment. For bar 2, we set up our new clean 11 tubes as it's shown in this table. Once placed in the required temperature as listed, we then took more readings using the spectrometer for every 20 seconds for 2 minutes. The final part of our lab was testing the pH. Our group did not do this part of the lab, but we did get the data from another lab that did to our calculation. For graph number 1, we are determining the effects of enzyme concentration versus the time. So our x-axis is the time and our y-axis is the concentration. And we are graphing 6 tubes. Tubes 2 and 3 are in blue, tubes 4 and 5 are in red, and tubes 6 and 7 are in green. And as the concentration increases, the time increases too. So we finally draw a straight line that best fits all the points for each condition of the tubes. In graph number 3, we are testing the rate of the reaction versus the temperature. So our x-axis is the temperature and our y-axis is the rate of the reaction. And when the temperature is so high, it causes the enzyme completely inactive. Then we draw the points, then we try to connect them, and we figured out that the maximum rate is 0.018 and the temperature is 50 degrees Celsius. In graph number 5, we are graphing the rate of the reaction versus the pH. Our x-axis is the pH and our y-axis is the rate of the reaction. And the pH affects the substrate binding efficiency of the enzyme. And we figured out that the maximum rate of the reaction is 0.01. I mean 0.011. And the pH is 4.
In the first part of the experiment, we found that 1 milliliter of the extract gave us the most linear line based on our data and graph. The line did not go up too fast and did not go too slow, letting us get the reading of absorption we needed, as shown on this graph in the middle line for the 1 milliliter. Using the 1 milliliter, we tested the absorbance in different temperatures. After graphing, we found the highest rate to be 48 degrees Celsius. Rate gradually went up with temperature, but when the temperature reached higher temperatures, the rate of reaction went down. I believe at higher temperatures the enzymes became denatured because of the hot temperatures, making them less effective. In the next part of our experiment, we tested the pH and rate of reaction on the enzymes. Once all the data was collected and a graph was made, it showed a trend that the pH didn't affect the rate of reaction much, except for when the pH was 3. I believe this pH was in the range in which enzymes cannot function for this specific enzyme, so it lowered the rate of reaction. Another possibility with our results could have been errors. Errors could have occurred in many locations. Contamination, adding the wrong amount of the solution, not enough of the solution, not enough time in the temperature selected, wrong pH added, could have all messed up our results. And thank you for watching.